Voice of the Sea, learning from experts across the ocean.
Welcome to Voice of the Sea.
In this episode, we're with University of Hawaii researcher Cecilia Hannides, defining the zooplankton food web from the surface to the deep ocean. Zooplankton are animals, many of which are small shrimp-like crustaceans that feed on algae and other tiny organisms. Zooplankton form the bulk of the diet of many larger fishes, and so Cecilia's findings are important to our understanding of how deeper water organisms are connected to food production at the surface, which is becoming increasingly important as commercial operations turn more and more toward harvesting deep-sea fishes for sale. We start on the research vessel Kilo Moana, where Cecilia and colleagues are collecting and processing samples for study.
I'm Cecilia Hannides. I'm with the University of Hawaii, and what I'm studying out here on the Kilo Moana cruise is zooplankton, which are small animals about 0.2 to 2 millimeters in size, mainly crustaceans like shrimp. And they're a very important part of the food web. They're kind of right between the particles that we're studying in the cruise. They consume many of the particles, and in turn they're eaten by many of the fish that we are collecting on the cruise as well. So I'm processing zooplankton right now, and we've collected zooplankton from 1,500 meters depth up to the surface of the ocean sequentially in 9 nets. So what I'm dealing with right now is actually zooplankton collected from 100 to 50 meters in the water column. They're, again, these very small organisms. What I'm doing right now is splitting the sample in half, and we're going to use the different parts of the sample for different analyses. This one is still filtering, actually. And I think do you want to look at what she's doing over there? Yeah. Okay. So Lauren's in the final stage of filtering sample onto mesh. We're actually vacuuming the zooplankton down onto a filter. So we have a vacuum trap set up with a 5-place filter rig, and we can vacuum all of the zooplankton in the sample from each sieve right down onto a filter. And then we actually weigh that back in the lab and have an idea of how much biomass or how much material is present.
This guy is obviously much longer than 2 centimeters or 2 millimeters, but he obviously fell through.
It's an operational definition.
Okay.
Now that the samples are collected, we follow Cecilia to her lab at the School of Ocean and Earth Science and Technology. Hello, you're watching Voice of the Sea. In today's episode, we'll be learning about zooplankton with Cecilia Hannides, a researcher at the University of Hawaii at Manoa. Thanks so much for having me here today, and we're going to learn about your research on zooplankton.
That's exactly right. So today we're going to be talking about zooplankton that I collected on cruises to the north of Oahu. We're interested in zooplankton. These are a very diverse group of organisms. They include things like Copepods and euphazes, which are crustaceans relative of shrimps, and many other different types of organisms, including jellyfish and arrowworms and chaetognats. We have a number of different types of samples to look at today. These are formalin-preserved zooplankton samples. They were collected on our August-September cruise. You can see differences between samples collected in the surface and those collected deep in the water column.
Oh, wow, there's a big difference.
There is a big difference. We collected these using nets, a special net system called the Mach Ness system. These nets collect zooplankton from quite large volumes of water. The surface water samples represent a volume of water about 700 meters cubed, which is about 185,000 gallons. The deep water samples are an even larger volume of water. These were collected from about 4,000 meters cubed, and that's over 1 million gallons of water.
So this is, in 1 million gallons of water, this is all that you collected, or this is a subsample?
No, this is approximately half of we collected from that volume of water.
There's not that much.
Yeah, they're relatively dilute.
No, this guy is pretty big. This fish in here, he's still zooplankton?
No, we do collect the odd fish or very large crustacean in our sample. So we're going to identify them probably at the level of genus. We're going to pick out as many of those specific organisms as we need for the isotope analysis, because many of these zooplankton are very small. They're quite microscopic. That means quite a few of those organisms. So in some cases, I might need 500 of one specific type.
And then you'd put them all together and that'd be the one sample?
That would be one sample, yeah.
And what do you do with that?
Once we have the organisms together in a sample, it's run through the same type of analysis that many other samples are run through in our lab. It goes through a chemical procedure where we isolate the amino acids from those samples and put them into a form which can be injected on our isotope ratio mass spectrometers. So we basically analyze the amino acids in these organisms for their isotope ratios. We're interested in this because the nitrogen isotope ratios are giving very interesting patterns that are indicative of utilization of surface water sources or deep water particle sources.
And so I see you have other samples out here. Can you tell me what's going on with these?
These are also samples that were collected by the same Mach-Ness net system, and they were treated in a different manner. Whereas in the latter case, many of our samples were formalin preserved, these were actually sieved live onto different mesh sieves and then filtered onto these filters. So what we have here are zooplankton filtered down from a 0.2 millimeter, a 0.5 millimeter, a 1 millimeter, a 2 millimeter, and a 5 millimeter sieve. So we have different size fractions of zooplankton.
They kind of look like freeze-dried vegetables.
That's exactly because they were freeze-dried. So we get them into the lab frozen, and then we weigh them for what we call wet weights. And then we actually freeze-dry them and weigh them again to get dry weights. As you can see, there's a lot more zooplankton on these top filters than on the bottom filters. Mm-hmm. And these zooplankton were again collected in surface waters in the 0 to 50 meter layer. And these zooplankton were collected down at the very bottom of our depth stratum, 1,000 to 1,500 meter layers. So this is from what we call the upper bathypelagic. And again, you can see that there's a lot more zooplankton biomass collected in surface waters, and we actually get very little zooplankton down at depth.
And does that mean that the deep part of the ocean is not as interesting or not as important as the surface if there's so many more organisms up at the surface?
Oh, no, not at all. We see very interesting trends in composition and biomass and in their chemical composition between the surface waters and the deep waters. And we think what that means is that the deeper zooplankton are actually consuming these suspended particles, which have a very heavy nitrogen isotope signature. So that's kind of one of the main outcomes of our project.
And those suspended particles are things that are kind of so tiny that people didn't used to think they could be important as food, is that right?
Right, they're very tiny. They're not only very tiny, but it was thought that they're actually fairly recalcitrant, so they are maybe not as edible as other types of particles like the sinking particles. So they're basically discounted as a food source. But from the results that we have so far, it looks like these suspended particles might actually form part of the food source for the zooplankton down at depth.
And that's really exciting because there's a lot of space in our ocean, right, that's that depth.
That's exactly right. This is a massive habitat for zooplankton across the globe. So what we're finding out here in the North Pacific is really applicable to zooplankton populations globally.
The University of Hawaii's Sea Grant College program, focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
the Curriculum Research and Development Group in the College of Education at the University of Hawaii at Manoa. CIDG's training roots go back over 40 years. Through professional development programs, curriculum workshops, research on teaching methodology, individualized school and district training, and so much more. The Curriculum Research and Development Group, improving schools, improving education. CIDG.
COSEE-Island Earth is working to establish new avenues for connecting research scientists with educators and communities. COSEE-Island Earth is working to enhance the science and ocean literacy of our island residents and visitors. COSEE-Island Earth is working to connect scientific research, traditional knowledge, and ocean policy. COSEE-Island Earth, bringing together university, government, research, and community partners to enhance ocean literacy and engage all ocean users in stewardship.
Welcome back. We're in the laboratory with Cecilia Hannides, looking at different sized zooplankton in order to better understand the ocean food web.
We have the zooplankton dried in a freeze dryer, and then they're ground, and they end up in a powder, like you see here, for again the different size fractions. And then these powders are weighed, and that is the samples that are then run for isotope analysis.
And do you literally grind them up with the mortar and pestle?
Yeah, this is an agate mortar and pestle, which is a very clean way to grind samples. And so we have many nets. For each toe, there's 9 nets on the mock nest and 5 size fractions for each net. So you can imagine this is quite a procedure to grind all of those samples. But that's exactly what we do. We take the samples, grind them in this mortar and pestle, then weigh them and analyze the sample.
It seems a little bit primitive in this high-tech lab for you to have the mortar and pestle.
Yeah, well, it's actually the best way that we have to grind the samples. Again, the agate mortar and pestle is very clean. It doesn't introduce any sort of contamination into our sample. And realize that we're dealing with many different types of samples. Sometimes we have a large amount of biomass, such as you would see in the surface waters. But sometimes we have really, really small samples that you would see, for example, in the zooplankton down at depth. And you have to have total control over that sample. This is the small size fraction of the zooplankton collected in the surface waters. And we just take the filter out.
Oh, you took the whole filter.
We take the whole filter out and we just gently scrape it off. So we want to get an idea of how much biomass, how much zooplankton material total there is at each depth, so in the surface waters. So we have those weights already. And now what we're doing is we're just going to grind up the sample so it can be analyzed for its isotope composition and its nitrogen and carbon content. So now we have the sample in the mortar. We're going to grind that.
And is there a certain technique you're using?
No, some of the samples are easier than others. This is actually a fairly easy sample because the organisms are small. I've gone to the smallest size filter, and you just grind it for about a minute or so. But again, we do catch much larger animals in some of our other nets and filters, and those can take quite a while to grind. We had some samples that we were grinding a few months ago that took over 30 minutes to grind to completion.
Oh my goodness.
And you want the powder to be homogeneous, basically, so that any subsample you take of this powder represents the entire sample.
I can smell it. It smells like a shrimp chip.
Yeah, that's exactly, most of the zooplankton here are small crustaceans related to shrimp, so that's probably exactly why. So once the sample is ground, we just take it out, carefully put it in a clean vial, and then it's ready for the next stage, which is weighing and then finally analysis.
Welcome back. We're looking at zooplankton under the microscope with Cecilia Hannides and her colleague Jeff Drazen.
Here we have some zooplankton sample. These were zooplankton that were preserved, and we're going to take a look at them under the microscope now.
And you literally count how many of each type or species that you're looking for?
That's right. So for the isotope analyses, we're actually looking for certain taxa, so mainly certain genuses of zooplankton. So we're looking for specific organisms, and what we do with that is we kind of go through the sample slowly, and when we come across the specific organism or taxa that we're looking for, we'll eventually actually take those out of the sample and then run the isotope analysis on those specific organisms from the sample.
Can you tell me the names of some of the organisms that you're targeting?
Yeah, we're interested in organisms that might have a link to these suspended particles that are found in the deeper waters, in what we're calling the midwaters or twilight zone, and also the upper bathypelagic zone. The organisms that we're looking for, some of them are what we're calling small resident particle feeders. A copepod down at depth called Oithona, potentially a different type of a copepod called Microscope. So that's one group of organisms that we're looking for, are these small resident particle feeders.
Can I take a look?
Yeah, definitely.
Oh yeah, I see some, those are copepods in there, right?
Yeah.
So right here, we're looking at part of a zooplankton net sample that was collected here off of Hawaii. And most of these things with long antennas that you can see right there, I'm pointing them out with the forceps, those are copepods. And they are what eats a lot of the microalgae in the ocean. We've got some other things in here. This is part of a gelatinous organism called a siphonophore. And these are arrowworms right here. And then this guy right over here, it looks a little bit like a small shrimp. That is a juvenile krill. And if we look through this sample, we'll see that there's mostly lots and lots of these long antennaed copepods. And they make up a large portion of the zooplankton in most of the world's oceans, actually. We've got some more of these little baby krill. Some of them might be baby shrimp. It's hard to tell when they're this size. And lots of different kinds of and different sizes of the copepods.
Now, can you
one right there.
put one copepod in the middle and we can just zoom in on him?
Absolutely. Let me find a big one. Oh crud, I got a predaceous one. This is Eukida. This is not an herbivore. See those big arms?
Yeah.
Yep. That's a predator.
What about that one in the corner there?
This one?
No, the other way.
This one? Or
no.
this one right here? That's, I think it's called Haptophilus, and it's a little unusual copepod, but it is definitely a grazer. Let me get the fish eggs out of there. So this is one of the copepods right here, and you've got these long antennae, and that's what it uses to sense the environment around it. And then these little legs coming off are what it uses to swim, and right in the middle, those appendages are clustered around its mouth, and that's what it would use to graze the algae out of the water column. And it's got a bit of a tail off the back side here, and that's a copepod.
Welcome back. We're looking at the results of Cecilia's chemistry analysis of her zooplankton samples.
These are samples which are being analyzed for their isotope composition. Right now we have the peaks coming out. We have some reference gas peaks. And what we're seeing here is a peak of nitrogen gas from the sample, and then a peak of carbon dioxide, the isotope ratio of carbon in our sample. This is basically where we get our results for what we're calling the bulk isotope ratios, the isotope composition of the entire zooplankton sample. You can see the peaks coming out, and then the trace of the isotope ratio above. We basically run all of our samples on this machine and get our bulk isotope compositions afterwards.
Can you tell by looking if that's a surface or a deep water sample?
Not necessarily, because we try to weigh out the same total biomass is put into each tin capsule, so we're hoping that we get approximately the same amount of nitrogen and carbon measured for each sample. In the bottom graph here we have the amount of nitrogen and carbon, and then the top trace is indicative of the isotope ratios, and eventually we'll get the isotope composition from those numbers.
Exploring Our Fluid Earth is a dynamic curriculum developed by the University of Hawaii's Curriculum Research and Development Group, teaching ocean science concepts through the disciplines of physics, chemistry, biology, and ecology. Exploring Our Fluid Earth is now available freely online. Find out more at exploringourfluidearth.org.
The University of Hawaii Sea Grant College Program, helping coastal communities of Hawaii and the Pacific through research, education, and outreach. Serving the community, from elementary to graduate students. Hawaii Sea Grant.